Okay, class. So we are back with another important lecture. And for the previous class and in the previous lecture, we have seen that what vascular bundles are and why there is need of the xylem and the phloem for the plants that we have got with us. Now, what our idea was that these xylem vessels that we have in case of the plants, so they were moving or they have to move the water enzymes all the way from the roots and into the stem, and ultimately they have to bring it toward the leaves because we know in the leaves, the water would be acting as one of the raw material for photosynthesis. So this means that our idea or our concept of the structure of the xylem vessels should be very much clear because we have to relate the structure of the xylem in context of its function that how come this would be able to move the water and the ions. Now let us forget the xylem vessels for a time for a time being and we see a situation from the daily life like if there was an underground water supply with me, and from this underground water supply, I have to carry the water with the help of a pipe to the houses that we have got at the top. So you understand is that I would not want any of the blockage coming here because if any of the blockage would come here, then that means that the water cannot be continually moved upward. And in order to provide this flat or this room with the surplus amount of the water, we have to make sure that these water pipes that we have got, they should be long and continuous, so they should be in the position to be moving the water over the long distances. So this is something that is also common, or we would see in case of the xylem vessel. So for the xylem, the first thing that we have to be considering is that these are the tissues. They are not the cells, they are rather than the tissues. And initially, when the xylem vessels in the plants would be developing, so it would be having some living cells in them. But later, when the xylem when they become mature, so the cells within the xylem they become dead. So this means that they lose the nucleus and the cell membrane and all of the stuff in them. And these xylem vessels they would be long and continuous. And why they have to be long? So that they can carry the water over long distances. And they have to be continuous, so at any point along the length of the stem, the water column must not break. And the other situation is that as I was quoting you in context of the pipe that was carrying the water from the underground water supply to the top of the room that we have got or the flat that we have got. So that pipe needs to have a wider diameter. Okay, so here you see that empty space that we have in the pipe. So this is a lumen. The same situation would be there in case of the xylem vessels as well. That these xylem vessels they are hollow or they are empty from the inside. And in these xylem vessels, we just have got these side walls, and these side walls they are made up of the cellulose, and later in their development, they would also possess these lignin in them, and that would provide an kind of an extra tensile strength to these xylem vessels. And for these xylem vessels, again, the situation is that there would be no presence of the cross wall. So this is a side wall, and if the wall is present like this, so this would be a cross wall. So the xylem they do not have these cross walls, but rather than they only have these side walls in them. So if I were just to show you the diagram that what I was talking about a while ago. So here you see I have got this handmade diagram for the xylem vessels. So here we have got these side walls of the xylem vessels. And on the inside, we have got the hollow empty space, and that would be regarded as a lumen. And you see that these are not the cross walls, but rather than this is representing the deposition of the lignin. And the lignin is not deposited initially, but later in the development of the plant. And this lignin would be providing a lot of tensile strength to these xylem vessels because when the water is carried in the xylem vessels under immense pressure, so there is a possibility that these xylem vessels can burst, but ultimately they don't because of the presence of the cellulose in the side walls and because of the availability of the lignin that we have in these cellulose. Now the situation is that how come this lignin is present within the xylem vessel? So this lignin can either be present in a spiral manner or this lignin can also be available within the xylem vessels in the form of the rings that I am drawing here for you. So this is how the lignin that we have in the plant. So this is how this is arranged within these xylem vessels. Now, if I were to show you another knee diagram of this, so if you see, like this is the side wall, and here we have got an empty lumen, and in this empty lumen only the water would be transporting, and these walls again they are later thickened with the lignin. You see, like here you see the lignin, this is being deposited in the spiral manner. So this is providing a lot of, you could say, the strength to the side walls that we have got in context of the xylem that we have here. Now, the thing is that we have to be relating the function of the xylem, we have to be relating the structure of the xylem to the function they would be performing. Okay, now if I were to consider a situation that here we have the xylem vessels, and in the lumen of the xylem vessel, if we had the living cells, so this would mean that there would be the presence of the cell membranes coming. There would be the cytoplasm coming, and you know that the cell membrane, they are the partially permeable membranes. So they would have resisted the flow of water. The water would not be able to be carried upward in a continuous manner, but the movement of the water would have got hindered if we had some living cells coming in the lumen. So that is why we say that the cells within the xylem, they are dead. And they would have no nucleus or the cytoplasm or the situation like that. And this has an advantage because by that the flow of the water is not restricted and there is nothing present in the way of the water that can provide the resistance to it. So the water would be moving without any much of the resistance. The other scenario is that since we have to be carrying the water over the long distances, and the long distance mean that from the root to the stem and then at the top to the leaf. So these xylem vessels, they are long and continuous, and the function that they would be rendering is that in this way, the xylem vessels, they would carry the water and ions over the long distances. The other situation was, as I have told you a while ago, that in these walls of the xylem, we have got the deposition of the lignin along with the cellulose. And when the lignin would be coming along with the cellulose, it would provide a high tensile strength to the xylem vessels, and these xylem vessels, they would not burst. The other situation was that these xylem vessels that we have with us, so they just have the side walls, they do not have any of the cross walls in them. So again, when there are no cross walls being present, so there would be no barrier to the flow of the water. And the inner empty space that we have in the xylem vessel that I was referring to this as lumen, so this lumen is large and wide. So in this way, we would be able to carry more volume of the water up the plant. So these are the points that are constantly being asked in the exam. That how come the xylem is adapted to be performing their function in context of the movement of the water and ions, and I have told you just now. Now moving onward, here we have to be discussing the structure of the other vascular bundle that we have with us, and these are entitled as the xylem vessels. The role of the xylem vessels is that they would move or they will translocate what? The sucrose that we have with us and the amino acids that we have with us. And if I were to tell you the structure of the phloems, unlike the xylem vessels, they are not dead, they are the living structure. And among the thing that we have we would have in the phloem vessels is that they have got the endoplasmic reticulums in them. The endoplasmic reticulum mean that these would be some membranes that would have the ribosomes in them, and there would be the presence of some fewer mitochondria in them. So they would not have any nucleus or the stuff like that, but since they have got this thin layer of the cytoplasm, the endoplasmic reticulum and the mitochondria, so they are living. And within the phloem vessels, which are a form of a tissue, so we have some individual cells and they are located in the form of a column that I will show you in a while. And these are referred to as the phloem cells. We also call them as sieve elements. We call them as the sieve cells. We also refer to them as the sieve tube elements. Then there would be coming up of the sieve and plate. We will see some sieve pores. Then there would be some companion cells present. And then there would be the availability of the side walls. And also we would have the cross walls in them. Like cross walls were not available within the xylem, but now they are available within the phloem. Now, here you have to be following me, and you see that if I were to be describing the structure of these xylem vessels, so we would say that these xylem vessels, they are the long tubes. See, they are the long tubes. And in these long tubes, we have got the column of these cells. So this is one cell which is in the form of a column. Now this is again another individual cell that I have got with me. And this is another cell that I have got with me. So here you see that this one individual cell that I am representing at the moment. So this is what we are referring to this as sieve tube element, or we call this as phloem sieve tube cell. This is here. And for every individual sieve tube element that I am showing you at the moment, so they have in them a thin layer of the cytoplasm, which is denoted by a blue marking. They would have some fewer mitochondria in them. And at the same time, they would also have fewer endoplasmic reticulums. And on those fewer endoplasmic reticulums, there would be the presence of these small spherical units, and they are the ribosomes. Now, another situation is that The cytoplasm of one phloem sieve tube element, this is connected to the cytoplasm of another sieve tube element, and for that we have got these pores in between the sieve tube element, and these pores, they are the sieve pores. The point where one sieve tube element it joins with adjoining sieve tube element, so there would be making up of a plate coming over here, and that plate it would be regarded as the sieve end plate. And in this sieve end plate, actually we have got these sieve pores that would connect the cytoplasm of the one sieve tube element to the cytoplasm of another sieve tube element that we have got. Now the problem is that since these sieve tube elements they do not have the nucleus in them, so the situation would be that they would require some assistance. Like if I were to quote you a reference from the daily life, so we have got our friends, and in some scenarios we also call them as companions. Why we call them as our friends or the companions? Because in the daily life, at some point, we get stuck where we need the assistance. So we say that, okay, we have got our friend, we have got our companion that is going to assist us. So the same way, these sieve tube element, they cannot survive alone themselves. So we need to be connecting these sieve tube elements with another cells, and these cells, they are the companion cells. So if you were to see here, in this diagram. So here I have got one sieve tube element, and this one sieve tube element would further be connected to a cell, and that cell is a companion cell. And if we see the structure of the companion cell, so we have got this nucleus in them, we have got the large vacuole in them, we have got abundant mitochondria in them, we have got the ribosomes in them. So this means that these companion cells, they are very important for the survival of the sieve tube element. They assist them in some functions, and that is why the sieve tube element that we have here, so they can survive without having the nucleus in them. Now we have concluded that these phloem sieve tube element that make the phloem vessel, so they would be translocating the sucrose and the amino acids. So from where do these sucrose and the amino acid would come? So this means that along with the sieve tube element and the companion cells, we have to be connecting them to the photosynthesizing cell. So here we have got a photosynthesizing cell, and this could be a palisade mesophyll cell, or this could be a spongy mesophyll cell. So overall, the idea is that the photosynthesizing cells, they would make the glucose and the sucrose and the amino acids. And these companion cells that we have with us, so they would be utilizing their abundant mitochondria to be releasing the energy in the form of ATP, and by that we would be moving the assimilates like collectively for the sucrose and the amino acid that we intend to be translocating from those part of the plant that are photosynthesizing. They are termed as assimilates, so they would come from here to the companion cell, and from the companion cell they would go into the phloem sieve tube element, and that phloem sieve tube element would now be moving these assimilates, the sucrose and the amino acid, to the other part of the plant where they are required. So if I were to quickly summarize this, this is phloem sieve tube element. Here we have the companion cell, and this companion cell is assisting the phloem sieve tube element. And this companion cell further is aligned with the photosynthesizing cell from where they would be taking up the sucrose and the amino acids. Now, here let me just try to summarize this quick. That what are the properties that we have got for the companion cell? So we say they have the nucleus, they have the cytoplasm, and then they have got the abundant mitochondria in them, right? Not the chloroplast, but rather we say that they have got the abundant mitochondria in them, and these abundant mitochondria that we have got, so they would provide sport, life support to the phloem sieve tube element. And those phloem sieve tube element later they would be translocating the sucrose and the amino acid. So if I were to tell you quick that what is the general, you could say, scheme in which we would be able to be moving the assimilates. So first in the scheme there would be the palisade mesophils and the spongy mesophyll cells coming. They would photosynthesize, would make the glucose and the sucrose and the amino acids as well, and then they would be carried to the companion cells, and from the companion cells we would now take them to the phloem sieve tube element. So if I were to quickly tell you about the characteristics or the structure of the phloem, that how this is related to the function. So we say that in the long phloem tubes that we had got these phloem sieve tube elements, the individual cell of the phloem vessel. So they have the thin cytoplasm and the fewer mitochondria. So this has an advantage because the resistance then to the flow of the sucrose and the amino acid would be minimized. Other than that, we know that the phloem vessels, they are accommodated by the companion cells, and they have the abundant mitochondria in them, and that would provide the life support to the sieve tube element and would protect them from dying. Other than that, we have seen that if I have got this one phloem sieve tube element and this is another phloem sieve tube element, so here you see that when they join with one another or they make a connection with one another, so here we were making a plate and that was sieve end plate. And in the sieve end plate, we have got the sieve pores. And when we have the sieve pores, so this means that they would act as an interconnection and would accommodate the movement of the sucrose and the amino acid from one phloem sieve tube element to the other phloem sieve tube element that we have. The other situation is that these phloem sieve tube element that we have got, so they have got their side walls like this. So here I'm making their side walls and they also have the cross walls in them. And their side walls that we have got, so they are again made up of the cellulose and they would provide the strength and the support to the phloem vessels overall. So this is what you have to be remembering, that how come the structure of the phloem is actually designed and how it is related to the function. So I hope you would be clear with these structures. And for the next class, we would see that how the phloem vessels they would be moving the sucrose and the amino acid up and down the plant.